So hello friends, in this video we will study about the biotechnology and its applications. This video is very important for NEET aspirants as we'll discuss very important chapter, that is biotechnology and its applications. This chapter is very important for the NEET examinations. We'll see various important points related with this chapter and we'll discuss everything in detail. Now I'll see what is biotechnology. So biotechnology basically deals with the industrial scale production of the biopharmaceuticals and biologicals using genetically modified microbes, fungi, plants and animals. The application of biotechnology includes therapeutics, diagnostics, genetically modified crops for agriculture, processed food, bioremediation, treatment and energy production. Now I'll see introduction of genetically modified crops. So there are various kind of applications like food industry, chemical industry. These are various kind of applications of the biotechnology. In agriculture also, then paper industry. Biotechnology is used in various fields. It is also used in environmental protection also. So these are some of the applications of biotechnology. So biotechnology applications in agriculture we'll see. Basically it is used to increase the production of the food, which is achieved by the agrochemical based agriculture, organic culture, genetically engineered crops based agriculture and green revolution. Green revolution is basically crop production is increased by using improved crop varieties, better crop management practices and use of agrochemicals. Now I'll see major research areas of biotechnology. To supply best catalyst, that is in the form of microbes or pure enzymes in the form of improved organism with better characteristics, to create optimal conditions by genetically engineered for a catalyst to function, and purification of proteins, organic compounds by developing downstream technologies for the isolation and purification of that organic compound. These are applications of the major application and research areas of biotechnology. So here you can see in the figure bacterial DNA is extracted, that is DNA extraction and isolation, then cloning and designing of the gene, then transformation, cell and tissue culture, then plant breeding. So different varieties of plants are produced by using this kind of bacterial genes and cloning and transforming and in fact basically used in the tissue culture. Now you'll see about the mechanism of Bt toxin and Bt cotton. So basically Bacillus thuringiensis, that is Bt protein, which kills insect like lepidopterans, that is tobacco, budworm, armyworm, coleopterans, beetles and diphterans, flies and mosquitoes. Bt toxin gene express itself protein crystal at the particular phase of bacterial insecticidal protein. So Bt toxin exists as inactive protoxin and on the digestion it basically perforates the insect gut lining. Now pest resistance plant. Biotechnology in agriculture basically helps in production of pest resistance plant. Development of pest resistant plant reduce the use of pesticide. One such approach is introduction of Bt toxin to target plant. Example Bt cotton, Bt corn, tomato, potato and soybean. Bt toxin produced by Bacillus thuringiensis due to presence of Bt toxin gene. So this is how Bt cotton is produced. Now I'll see in the figure you can see there is Bt sugar is there and Bacillus thuringiensis is present. Now it is ingested by the insect. So basically, after ingestion, it solubilizes and converted into protein crystal converted into protoxin, and it activates, and there is delta toxin is produced, and these toxins insert into the cell membrane of the gut lining. So cryotoxin bind to the midgut receptor and it create perforations. Now I'll see the different kind of genetically modified food produced by the biotechnology like soy, corn, tomato, which is also called pomato, which was developed by biotechnology. Then potatoes, different genetically modified papayas, peas, dairy products, cotton seeds, rice, and canolas. These are some of the top 10 genetically modified food produced by the biotechnology. So these are some of the examples. Now I'll see in this figure how BT cotton is produced. Basically, BT inactivated form of the uh, sprayed on the corns. So eaten by the insects, toxin get activated by alkaline pH of the insect gut and swelling of the gut of the insect, and basically there is death of the insect as you can see in the figure. So this is how BT crops work. They mainly get activated inside the insect gut and it create perforations inside the insect gut so that insect gets killed. Now melodogene incognitia. This is also an important topic in the NCERT. Basically, this here, a nematode is basically killed by a technique called the RNA interference, where its protein production by this melodogene incognitia is inhibited with the help of the RNAi technology, where a single RNA is inhibited by its complementary strand sequence. So here you can see method to genetically modified crop through RNAi. Vector use agrobacterium. This is natural genetic engineer of plant to introduce nematode specific DNA. So nematode specific gene introduced into the host plant, transgenic plant. DNA will produce both sense and antisense RNA in the host. Sense and antisense RNA are complementary to each other, double-stranded RNA initiates RNAi. So basically, sense strand is that strand which is producing protein. So it is inhibited by this RNAi. So pest resistant plant. BT toxin is not toxic to all the pest. It is very specific or pest become resistance against BT toxin. Another approach to develop pest resistant crop against nematode parasite is RNA interference as we have discussed. Example, melodogene incognitia, that is in fact mainly root of tobacco. So principle of RNAi is to develop pest resistance by silencing translation of specific mRNA. RNAi takes place in all eukaryotic organism as a method of cell defense. So this is also a very common phenomenon. So this figure you can see there is double-stranded DNA and there is dicer binds and cleaves double-stranded RNA into dicer binds and cleaves double-stranded RNA into siRNA and direct introduction of siRNA into the cells. So this the siRNA is unwound. The RNA induced silencing complex that is called RISC and cleavage of the target mRNA is done. So this is the mechanism of the RNA interference. Now I'll see biotechnology applications in the medicine. Biotechnology in medicine is mainly used to diagnose and treat different diseases with effective recombinant therapeutic drugs, protection from dangerous diseases through gene therapy, and new health genes can be inserted into replace gene damaged cells. Target action of the drug by thorough study of the genomics. And recombinant therapeutics do not induce unwanted immunological response as in case of similar product isolated from non-human sources. So these are some of the applications. Now I'll see genetically engineered insulin. So deficiency of insulin causes diabetes, which can be controlled by taking insulin at regular intervals. Traditionally, when humans insulin were not available, insulin of animal like slaughtered cattle and pigs were used. Animal insulin triggered some immune response like allergy and some reaction due to foreign insulin and will not be effective. So bacteria were genetically engineered to produce human insulin in large quantities as bacteria can easily grow in the culture medium. So genetically engineered insulin, first created in 1983 by Eli Lilly, an American company, insulin hormone to regulate blood sugar level and administered in patients with diabetes. Mature insulin, two short polypeptide chain that is chain A and chain B, which are linked by disulfide bridges. Insulin synthesizes prohormone in the all mammals. A peptide B and additional C peptide are sent in mature insulin as it get removed during the maturation. Using rDNA technology, two DNA sequences corresponding chain A and B insulin introduced into the plasma of E. coli cells. So this is how genetically engineering of insulin is done. So this synthesized DNA encoding B chain. Gene for B chain is cloned and B polypeptide is there. And synthetic DNA encoding A chain is there. Gene for A is cloned and A polypeptide is produced. And these two chains are combined with the help of the disulfide bond by purification and mixing A and B chains to produce the genetically engineered insulin. So maturation of proinsulin to insulin. So there is proinsulin and B cell peptides, and insulin is produced by combining A and B chain with the free C chain. So this is how maturation of proinsulin to insulin takes place, as you can see in the figure. Proinsulin is basically an inactive form of insulin, and when these two chains combines with the C, it produces the active insulin. Now, what is gene therapy? Is a corrective therapy to correct or treat hereditary diseases, a gene defect diagnosed in the child or embryo. Method where genes inserted into a cells and tissue to treat disease. So principle of gene therapy is mainly normally a gene when get inserted into individual cell or embryo with gene defect, the normal gene will take over the function of and compensate for the non-functional gene. So first clinical gene therapy was given to 4-year-old child suffering from ADA deficiency. So method to treat ADA through gene therapy. So basically adenosine deaminase deficiency, this is an enzyme disorder, hereditary disorder caused due to deletion of gene for adenosine deaminase. Enzyme adenosine deaminase is crucial for the immune system function. In the gene therapy, lymphocytes from blood of patients are isolated and grown in culture medium. Functional ADA-cDNA introduced into lymphocyte through retroviral vector and returned to patients. Since lymphocytes have short lifespan, genetically engineered lymphocytes are periodically introduced into patients and it cures the patient. So this figure you can see, first is cell harvested from the patient, second is into the lab virus altered so it cannot reproduce. Third is a gene is inserted into the virus. Fourth is altered virus mixed with patient cell. Fifth is cell becomes genetically altered. And sixth is altered cell injected into patient's body. And seventh is altered cell produces desired protein. So this is how gene therapy works in case of ADA deficiency disease. Now I'll see how biotechnology helps in molecular diagnosis. So techniques used to diagnose and monitor disease, detect risk and detect which therapies will work best for the individuals, patients. Treatment of a disease be effective, early diagnosis, understanding physiology very important. Conventional method of diagnosis like serum and urine, early detection of disease is not possible. Early detection possible with advanced molecular diagnostic tools like recombinant DNA technology, polymerase chain reaction, enzyme like immunosorbent assay. So this is how biotechnology helps in molecular diagnosis. Now we see significance of molecular diagnosis. So molecular diagnosis like PCR detect pathogen even at very low concentration. Amplification of nucleic acids in involved. That's why detection of HIV, immunodeficiency virus in suspected AIDS patients, and detected mutation in gene in suspected cancer patient. Detect many genetic disorders. PCR requires single standard DNA or RNA tagged with radioactive molecule to hybridize complementary DNA in clones. So these are the significance of molecular diagnosis. Now it is another application is of biotechnology in molecular disease diagnosis, ELISA. Other molecular diagnosis detect antigen, foreign substance or pathogen, works on the principle of antigen antibody interaction. ELISA detects pathogen by the presence of antigens, proteins, glycoproteins, etc., or by antibodies synthesized against the pathogen. So it will give a uh, different colored intensity according to the presence of the antigen in your sample by a plate reader, as you can see in the figure. Now PCR. What is the principle of PCR? So there is a clinical sample and DNA preparation, amplification of the target DNA, and there will be a probes. The DNA will be denatured and it will combine with the probe, and there will be detection of your particular gene involved in disease. So this is how PCR is used in the diagnosis of different kind of diseases with the help of the probes. Now I'll see transgenic animals. May be defined as animals that have their DNA manipulated and possess and expresses a foreign gene known as transgenic animals. So as you can see in the figure, there are various kinds of transgenic animals here, like pig, cow is there, different kind of genetically modified rats and mice are used in lab. So these are transgenic animals which are used in different kind of experiments. Now we'll see reason to produce transgenic animals. Normal physiology and development. Transgenic animals are designed to study different aspects of gene, like how genes are regulated, how they affect human function of the body and its development. Example, study of insulin-like growth factor involved in growth can done by introducing gene from other species that alter the format of formation of their factor and study the effect of the that results also in the formation, also about biological role of factor in the body, such as just an example. No vaccine safety. For vaccine safety, vaccines are firstly tested in transgenic mice, developed for use in testing before they are used in humans. Transgenic mice are being used to test and safety of the polio vaccine. If successful and found to relatable, they could replace the use of the monkey to test the safety of batches of the vaccine. Chemical safety and testing. Transgenic animal introduced with genes which make them more sensitive than non-transgenics. They are then exposed to toxic substances which affect studied. Now, see ethical issues. So, manipulation of the living organism by the human race requires certain regulation. Ethical standards are required to evaluate the morality of all human activities that might help or harm living organism. Genetically modification of the organism has unpredictable results, which such organisms are introduced in the ecosystem. Indian government has set up organization like GEAC, Genetic Engineering Approval Committee, which will make decisions regarding the validity of GM research and safety of introduction of the GM organism for public services. So these are the some of the genetically modified organisms. As you can see, there is a ear grown on a mice. So these are the lab animals which are used for the different purposes and maintained. And whenever someone requires to do a particular experiment, they can choose an organism and just take an approval from the institutional ethical committee and start the experiments. Now about 2 lakh varieties of rice found in India, which makes India richest. Basmati rice, one of the type rice which unique aroma and flavor. And there are 27 documented varieties of basmati rice grown in India. 1990s have been made over 2000 variety of rice had been found in India, which makes India richest. Basmati rice is one of type of rice with unique aroma and flavor. There are 27 documented varieties of basmati rice in India. In 1997, an American company got rights to basmati rice. So, use of genetic material, plants, and biological resources by companies granted with patent has triggered public and anger since they have been identified, developed, and used by farmers and people of specific country and society. So that's called biopiracy, and it is uh, very bad because if you will copy and just use it for your benefit, the traditional knowledge used by farmers, then it will be very lossful process for them. Now, several attempts has been made to patent uses product based on Indian traditional herbal medicines like turmeric, neem. We should be vigilant and counter these patent applications, else other countries individual may clash on our legacy and rich legacy, and we may not be able to do anything about it. Okay, so that's why protecting our traditional knowledge of herbs and medicines are important so that other companies cannot misuse it in the form of the biopiracy. Now I'll see what are there are number of biosafety protocols the story so far, that is patent office. There is so as you can see in the banner, it is written rice is life, not corporate business. So no to GMOs. That's why you need to protect the different crop varieties like basmati rice, which they have remained and started selling in the name of Basmati and Texamati. Because basmati rice mainly belongs to India and its patent should be remain with India. Now you see relation of the injustice, inadequate compensation and benefit sharing between developed and developing countries led to some nations to develop laws to prevent such unauthorized exploitation of bioresources and traditional knowledge. The Indian Parliament has cleared the second amendment of the Indian Patent Bill that takes such issues into consideration, including patent terms, emergency provisions, and research and development initiatives. So these are the some of the measures taken against the patent granted unlawfully. Now, so the patent mafia that is doing the biopiracy must be stopped for the Greenpeace. As you can see, this figure shows that only. So this biopiracy is a very bad thing because it just takes the natural resources and the nature's gift from one place to another place and just exploit it for its own profit, and native people does not get anything from that patent. Biopiracy, the patenting of plant genes and other biological products that are indigenous to another country. Biopiracy use of bioresources by multinational companies and other organizations without proper authorization from the countries and people concerned without compensation payment. Developed countries patent the knowledge and resources of underdeveloped countries and enjoy immense profit. Industrialized nations are rich financially but poor in biodiversity and traditional knowledge. That's why, in contrast, the developing and underdeveloped country world is rich in biodiversity. So they exploit the developing countries. Now I'll see. So likewise, these are the some of the things which are important for the NEET examinations from the chapter like biotechnology and its application, and it is very important chapter. So this is a one short video. I hope this video helps. So kindly subscribe and give it a thumbs up. We need your support, and I'll bring all the video series with one short videos for you of each chapter. Kindly give it a like so that I can be motivated to make such kind of videos.